This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Okay, welcome everyone. I'd just like to start off acknowledging the traditional owners of the unceded land on which we work, learn and live, the Wiradjuri, Woiwurrung and Bunurong people, the Yorta Yorta Nation and the Jar Jar Wurrung people. We also acknowledge and are grateful to the traditional owners, elders and knowledge holders of all Indigenous nations and clans who have been instrumental in our reconciliation journey. We recognise the unique place held by Aboriginal and Torres Strait Islander peoples as the original owners and custodians of the lands and waterways across the Australian continent, with histories of continuous connection dating back more than 60,000 years. We also acknowledge their enduring cultural practices of caring for country. We pay respect to elders past, present and future and acknowledge the importance of Indigenous knowledge in the Academy. As a community of researchers, teachers, professional staff and students, we're privileged to work and learn every day with Indigenous colleagues and partners. So thank you. Now, today we have a confirmation seminar presented by Shijie Geng. For those of you who have not met Shijie before, he comes to us from southern China. And sorry, I'm just trying to get my, where have I lost it? I've lost my introduction to you, Shijie. But he completed a Bachelor of Animal Science at South China Agricultural University in 2016, and then a master's at Shijie Geng University, and then worked at the Chinese Academy of Sciences at Guangzhou for 2 years as a research assistant. He joined the team late last year, initially offshore, but luckily was able to join us pretty quickly after starting his PhD. And he's going to tell us today about his project, which is focused on identifying essential gene functions responsible for the survival of Mycoplasma bovis in cell culture. So please take it away, Shijie. Thank you.
Thank you, Glenn. Good morning, everyone. Welcome to my PhD confirmation seminar. I'm really honored to have this opportunity to present my proposed PhD project, which is around the identification of essential gene functions responsible for the survival of Mycoplasma bovis in cell culture. I'm Shijie, and I'm now working with Glenn, Nadika, Kelly, and Sarah. And my committee chair is Joanne. I will start with an overview of the background information of my proposed project. So according to the taxonomy of mycoplasmas, they are bacteria under the class molecules and are important pathogens in humans, animals, insects, and plants. In humans, for example, Mycoplasma pneumonia is a causative agent of respiratory infection, while in livestock industry, Mycoplasma bovis, Mycoplasma gallisepticum, and Mycoplasma synovia are representative mycoplasmas causing respiratory diseases. Here are some major characteristics of mycoplasmas. They are the smallest bacteria with the smallest genome. They also lack many biosynthetic and metabolic capacities due to reductive evolution from gram-positive bacteria. Therefore, they are highly dependent on the incorporation of nutrients from the host cells. They also have variable surface proteins, which can help them evade the host immune system. And there is also horizontal gene transfer, which results in acquisition and transmission of chromosome segments between different strains and species. I'm now working on Mycoplasma bovis, which is one of the most economically important pathogens in the livestock industry. It causes chronic diseases in cattle at all ages, including mastitis, pneumonia, and arthritis. It is also associated with meningitis, cardiac diseases, and reproductive diseases. The left figure here shows the arthritis in a calf infected with Mycoplasma bovis. We can see swollen and inflamed couple joint, and it is also discharging yellowish material. The picture in the middle shows the lesions of Mycoplasma bovis pneumonia with multifocal white nodules elevated on the lung. And the right picture here shows the colonies of Mycoplasma bovis isolated from the affected lungs. The genome of Mycoplasma bovis has been fully analyzed by whole genome sequencing. However, the annotation of many genes is still based on prediction and remains to be fully illustrated. We can see that the genome size of Mycoplasma bovis is around 1 million BP, and there are 6 VSPs identified in the HB0801 strain and 13 VSPs identified in the PG45 strain. Given the huge impact of Mycoplasma bovis on animal production, many diagnosis methods have been developed for detection of this pathogen. Culture is still the gold standard and the primary method, but it is time-consuming and the work is laborious. In order to solve these problems, PCR and qPCR methods are developed for quick detection, but they are still highly dependent on culture enrichment and facilities. And there are some other diagnosis methods, such as multiplex qPCR for detection of multiple pathogens, loop-mediated isothermal amplification, which is really user-friendly and time-saving, and also mass spectrometry-based diagnosis method for the detection of viable cells. There are also commercial ELISAs for Mycoplasma bovis, including indirect ELISAs, which target Mycoplasma bovis antibodies, and sandwich ELISAs, which target Mycoplasma bovis antigens. Antibiotics have been used to treat Mycoplasma bovis infection. Although lack of a cell wall means that this pathogen is naturally resistant to commonly used antibiotics, such as penicillin and polymyxins, there are still antibiotics which are active against this pathogen. However, the antibiotic treatment is increasingly less effective. This is a research investigating the susceptibilities of 62 Mycoplasma bovis strains isolated from cattle in Britain. And they have used minimum inhibition concentration and minimum mycoplasmacidal concentration to illustrate the shifts in susceptibilities of the Mycoplasma bovis population to antimicrobials. We can see that fluorophenical and spectinomycin are less effective in the treatment of Mycoplasma bovis. And this pathogen is completely resistant to tilmycin. The increasingly limited efficacy of antibiotics and antimicrobial resistant problems have raised the importance of conducting biosecurity practices. Here are some commonly used practices, including heat treatment or pasteurization to prevent spreading through milk and colostrum to cows, and also separation or culling of the infected cattle, as well as eradication by culling infected herds in New Zealand. Another strategy to control Mycoplasma bovis is the usage of vaccines. However, there are only 3 commercially available vaccines, including 2 bacterium vaccines, which are available in the United States, and also 1 attenuated live vaccine, which is developed by our team. There are also some other possible vaccine candidates that have been studied, but they vary in protective effects. Although some vaccine candidates have increased immune response in the host significantly, no significant difference was found in lung lesion and survival rate in some cases. So one reason we lack effective vaccines is the difficulties in applying genetic manipulation techniques for functional genomic analysis and also vaccine development. However, different genetic manipulation techniques for mycoplasmas have been developed in recent years. The transposon mutagenesis is the most commonly used method for random disruption of genes. And there are some other methods that can target specific genes, including recombination methods and also the CRISPR-Cas9 genome editing system. For my project, our lab has established a mutant library by successful application of the transposon mutagenesis for Mycoplasma bovis PG45. The left figure shows the experimental procedure of transposon mutagenesis. First, the mycoplasma cells need to be transformed with transposon plasmids. Then the transposon elements will be integrated into the mycoplasma genome by the induction of transposes. The right figure here shows the location of 319 transposon integration sites, which are distributed randomly in the Mycoplasma bovis genome. We have also done co-culture assay for the transposon mutants. So co-culture assay is actually an in vitro infection model to understand the interaction between pathogens and host cells. This picture shows the experimental procedure of co-culture assay for Mycoplasma bovis with MDBK cells. So MDBK cell line is actually a bovine epithelial infection model used for Mycoplasma bovis. The cells need to be inoculated with transposon mutants respectively and incubated for 3 days. The culture system will then be used for titration to determine the viable Mycoplasma bovis cells. And the titration results will be further used to calculate the doubling time of each mutant to identify the mutants that can or cannot grow with host cells. Previous functional genomic analysis is commonly divided into 2 steps. Bioinformatic analysis was used to identify homolog of the coding sequence to predict its putative function. Then the recombinant proteins were expressed for further in vitro assays to verify their functions. So the left figure here shows the determination of the polynucleotide binding capacity of a Mycoplasma gallisepticum protein, MSLA. The polynucleotides were synthesized with fluorescein labels and incubated with recombinant proteins. The products were further separated in gels, and we can see the polynucleotides that bind with recombinant proteins have reduced mobility in the gels. The right figure here shows the identification of the lipid binding property of a Mycoplasma bovis protein, MILA. So different kinds of lipids were incubated with the recombinant proteins on a membrane, and the proteins that can bind with the lipids were retained and detected by antibodies on the membrane. As a novel functional analysis technique, metabolomics has popped up on the scene in the past few years. Metabolomics is a mass spectrometry-based technique to capture and measure many metabolites simultaneously, including gas chromatography, MS, and liquid chromatography, MS platforms. So for Mycoplasma studies, it can provide us with a complete picture of the metabolic processes in Mycoplasma cells. And it can also give us a direction for further verification of the gene functions. So the right figure here shows a standard data set of metabolomics. Figure A and B are a heat map analysis and principal component analysis, which are used to identify the differences in metabolic profile of different samples. Figure C is the identification of significant metabolites, and figure D is called pathway analysis, which can map the significant metabolites to specific database for identification of significant pathways. Here are some applications of metabolomics for Mycoplasma studies. So one study used metabolomics to determine the function of the MARF gene in Mycoplasma gallisepticum, and it verified glycerol transport regulated by this gene. Another study is a comparative metabolomic study for Mycoplasma bovis and Mycoplasma gallisepticum, and it determined significant differences in metabolic pathways of 2 Mycoplasma species. In summary of the background information, there are still 3 problems facing the treatment and control of Mycoplasma bovis, including limited efficiency of antibiotics, the problem of antibiotic resistance worldwide, and inefficient protective effect of vaccines. Therefore, we still need to have a better understanding of the pathogenesis and virulence factors of Mycoplasma bovis. However, although the genome information of this pathogen has been fully reviewed and analyzed, more functional genomic analysis is still needed to determine the functions of the genes. Given Given the importance of metabolic functions of Mycoplasma bovis in the host cell interplay, this project aims to reveal potential metabolic functions of essential genes responsible for the survival of Mycoplasma bovis, which may provide insights for the development of novel vaccines. So the next section is around my research plan. My first aim is to determine the metabolic functions of the essential genes for survival in host cells. The essential genes identified in the co-culture assay will be used for bioinformatic analysis to find homologs and potential functions. For example, BLASTP and FIR2 programs will be used to identify homologs of the coding sequence, while BioSynthetic program will be used to identify potential metabolic pathways involved. And 3D ligand site can be used to identify the 3D structure and potential binding sites of the proteins. The candidate mutants selected using bioinformatic analysis will be used for further metabolomic detection to determine the alteration in the metabolic profile of the mutants. I will also compare the metabolomic results with the bioinformatic analysis to select certain metabolic functions. And for verification of specific metabolic functions, stable isotope labeling will be used to track specific nutrients through their metabolic pathways. For the bioinformatic analysis, 7 out of 12 mutated genes have been predicted to have probable metabolic functions, including D-galactose degradation, cholesterol transport, and also carbohydrate transport. 6 mutants have been selected for further analysis according to their probable metabolic functions and the prediction of transmembrane structure. In order to collect Mycoplasma cells actively metabolizing, growth curve experiments were conducted to identify the mid-log phase of growth of its strain. The parent strain PD45 and also one of the mutants 300 with a dilution of 1:60 and a dilution of 1:40 were used to optimize the experimental conditions. The cultures were incubated in 37-degree incubator, and 300 microliters of culture were used for titration every 2 hours between 12 and 24 hours to determine the concentration of the Mycoplasma cells. My results showed that the concentration of both the parent strain and the mutant reached the top around 14 hours, and 12 hours were selected as the mid-log phase for these two strains. Therefore, the dilution of 1:40 and the time points between 10 and 18 hours were used for my actual experiment. So this figure shows the growth pattern of all strains, and the growth curve experiments were also repeated for verification of these time points. In the end, 10 hours, 12 hours, and 14 hours were selected as the mid-log phase for the mutants and also the parent strain PD45. After collecting Mycoplasma cells at their mid-log phases, the cells were further quenched and washed, metabolites were extracted, and GC-MS detection was conducted. For now, I have received the data metrics for 6 mutants and also the parent strain PD45, and I'm still working on the analysis process. So my future plan is to identify the significant metabolites and pathways from the GC-MS analysis, and maybe conduct further metabolomic detection, such as LC-MS, to cover more metabolites that we are interested in. I will also use isotope labeling to track the metabolites through their pathways to verify metabolic functions. My second aim is to express and purify the proteins coded by mutated genes and perform further verification by functional analysis. For the expression part, target genes will be selected from the metabolomic study, and cloning and expression of the target genes will be conducted, and the recombinant proteins will be verified by Western blotting. For the functional verification part, recombinant proteins and its potential target nutrients will be used to demonstrate the enzymatic or metabolic capacities of the proteins. So for example, this study verified the dinucleotide degradation capacity of one recombinant protein in Mycoplasma bovis. The dinucleotides and also the nano-RNAs were incubated with the recombinant proteins, and the reaction system was detected by LC-MS. The peaks in this picture with the same elution time represent the same metabolites, and the presence of the nucleotide AMP verified the dinucleotide degradation capacity of this protein. My third aim is to investigate the potential role of metabolic pathways in the survival of Mycoplasma bovis in cell culture condition. As these mutants were unable to survive in cell culture with disruption of specific genes, I aim to further investigate the importance of these metabolic pathways for the survival of Mycoplasma bovis in cell culture condition. The mutants will be transformed with plasmids containing the target genes to restore their metabolic functions. And then the host cells will be inoculated with different strains, such as the parent strain PG45, the mutants, and also the mutants with restored metabolic functions. A growth pattern of these strains will be used to identify whether the restoration will reverse the abrogated growth in the cell culture condition. The cell culture media will also be supplemented with essential metabolites to investigate the importance of the metabolic pathways for the survival of Mycoplasma bovis. In the end, I would like to thank my supervisors, Glenn, Nadika, Kelly, and Sarah, and my committee chair, Joanne, for their supervision in my PhD study. Many thanks to Anna and Chinsa for helping me get familiar with the lab. I would like to thank Ling for her instructions on the Metabolomic experiment, and Dave, Nadim, Jordi from the MA team for their help in metabolomic detection. Many thanks to the members of the Mycoplasma Lab and Building 400 for your warm welcome, and the Melbourne Research Scholarship for providing me financial support in my PhD study. Special thanks to my wife Guihua for her encouragement and support in my everyday life. That's it. Many thanks to everyone for your attention.
Thanks very much, Shijie. Maybe keep slides there in case somebody wants to ask you specific questions about them.
Yeah.
Does anybody have any specific questions? Or any questions for Shijie? So I'll just look at the chat. But I think you can probably, there's not that many people here, so you can probably just unmute yourself and ask your questions if you have any. People, there's lots of congratulatory messages coming through. No questions as yet. Maybe I'll kick off while people are thinking, Shijie. In your growth curves, there were some differences between the different strains. What do you think might be going on there?
Yeah, there are different growth patterns for different mutants. I think because the mutants, because there are different genes mutated in different mutants, and these genes might play an essential role for the survival of Mycoplasma bovis in cell culture. So when they are growing in Mycoplasma bovis media, some of the functions, so they might lose some of the functions. And for example, if some genes are associated with nutrient incorporation, and if this gene is mutated, then this mutant has lost this function. So, but we can see that the mutants can still grow in the media, which means that the Mycoplasma cells might be adapting themselves to alternative nutrients for their growth. So, and a really interesting strain is the 0028, as you can see in this picture, because the concentration of this strain has already reached the top around 10 hours, which is pretty different from the other strains. So as you can see in this picture, there are the concentrations of these mutants are still going up and down. So I think it's really interesting to have a look at the metabolic profile of 0028 around 10 hours. Yeah, so we might find some interesting functions associated with the nutrient incorporation or things like that. That's what I think.
That's great. Thank you. A very polished presentation. So thanks very much. I think you'll get lots of virtual claps. So yes, I think that's it. So thank you very much. Thank you, Shijie. Thank you everyone for attending.
Thank you, everyone.
Shiji's confirmation presentation. I'll just stop the recording.
Yeah.